All right, I think we're going to get started here. Well, happy Friday, everybody. I'm Emily Pietzu. I'm a genomic educator at the Jackson Laboratory, and this is our second edition of JAX Career Chats. And I'm so grateful to be joined today here by Sarah Holbrook. And she is a graduate student at the University of Maine. She's doing her research in the Cox Lab at the Jackson Lab, and we'll talk about that a little bit more. But I just wanted to say welcome, and we're also, it looks like we have another guest joining us here. Sarah, who's that you have with you?
He's Crowley, the Crested Gecko.
Crowley,
he's my little buddy.
Gecko.
So he's very chill.
Wow, what a charmer. Well, thank you for joining us, Crowley, and thank you, audience, for joining us. Don't forget, if you have any specific questions or comments that you want to give us during the talk, feel free to give us those and we will integrate them into our talk as best we can. But why don't we just get started, Sarah, if you're ready. Maybe you can just give us a little bit of information about how you got to the Cox Lab in Bar Harbor, Maine, kind of where your path started and kind of how you started studying what you study today.
Yeah, so in the Cox Lab, so I'm a grad student in the Cox Lab. It's my third year. We study childhood neuromuscular degenerative diseases like spinal muscular atrophy, respiratory distress, and even like ALS. But how I got there is, it's a story. So I'm originally from good old northern Maine, from around the Presque Isle, Fort Fairfield area. So mostly potatoes and moose.
Oh, wow. So that's up near Canada, huh?
Yep. Yeah. I could see the border from my house. The sun would rise above Canada every day. So there was like a river out back, a potato field next door. It's very, very Maine.
Wow. So that's where you grew up and went to high school?
Yep. Yeah. And high school was, well, no, even in like younger childhood, I always kind of liked observing animals, like Crowley the Crested Gecko, or the moose and fox that would be outside. And I was really inspired by Steve Irwin to like really start looking at nature and asking questions and just really appreciating what's around me. But it was in high school with my biology teacher, Mr. McCrae. We actually went to Jackson Labs for a field trip, and we got to see some of the mice. One of them being the obies, so the obese mice. Their nickname was Hockey Puck. They're very cute. And then there's also the glow-in-the-dark mice. So you put a black light over them and they would glow green. And as soon as I saw them, I'm like, this is what I want to do. And I also wanted to be at JAX too. Like, so it's been a dream actually being here.
Wow.
But to
so
actually
you
get
actually
to JAX,
knew that you wanted to come to this place for a while because you had an experience that cool in high school.
Yeah, my biology teacher was great. Like, he'd always have extracurricular programs and after school activities. Like, he helped me go over the nervous system of a cat. Like, we also, yeah, and he set the desk on fire one time. That was good.
So very passionate educator.
Yeah, like he really, really inspired me. Like, really kind of grew that spark I already had for like the science. But in undergrad, I went to UMaine for biology. Okay. And I had so many different experiences. Like, I worked in a chytrid lab. So a chytrid is a type of fungus that has like a flagellum. It's known for, or at least Batrachochytrium dendrobatidis is known for killing frogs, especially in South America.
Oh, it's a pathogenic fungus? Cool.
Yeah, like it was very cool to study and just see its life cycle. And I think that was the first lab I officially went into. So I've always kind of had an interest in neuroscience, but I took that opportunity to go into the fungi lab because the neuroscience lab I was going to join, the Rosenwasser lab, which studies alcoholism in mice, there had to be some construction during the summer. So I'm like, I still want to do some sort of science. And even though fungus was like something entirely out of my realm, like I knew very little about it, I thought it'd be a great opportunity, and I got to work in the field. So I got to go out in bogs and collect field samples and try to grow and preserve these guys. And I gained a lot of valuable skills of just knowing like how to keep a sterile lab environment and how to use the microscope. But then I try to think, yeah, so then I got into the alcoholism lab where I studied mouse behavior, like male mice, especially when they get some alcohol in their system, become belligerent. They're just kind of tipsy and like
That doesn't sound
ridiculous
unlike
creatures.
human beings.
Yeah. And that like part of me was like, this research is very translatable. Like, and I also worked.
So you started studying alcoholism in mice during undergraduate at UMaine.
Yep.
Oh, okay.
Yeah, it was a great experience. I'm like one step closer to JAX. And, I even was able to conduct my own experiment looking at brain tissue and how alcoholism impacts it, like inflammation wise. And that was a collaboration between a bunch of different labs. And I really got to ask my own questions, learn how to use a cryostat, which is scary because it has like a very sharp blade. And, I'm still scared of it to this day.
Wow, that's awesome. So it sounds like you got to have a little bit of an independent research experience during your undergraduate with that project.
Yeah. And I was only really able to get those experiences because I took every opportunity I could to, well, basically network. Like I talked to my professors and made sure to ask questions, even if they seemed kind of silly. At UMaine, the biology classes had like 300 people in each section. And from Northern Maine, I had 30 students in my whole class. So it can be hard to like make yourself kind of stand out. So I think originally I wore a Stephen King hat, like reading Stephen King. And the professor first knew me as like, you with the Stephen King hat, what is the answer to this question? And eventually he found he knew my name, but because I would still ask questions and stuff like that too. But
that's really
if
good.
I didn't make those connections, even with other students who worked in other labs, I wouldn't have been able to do all the stuff that I did in undergrad and even grad school now. Because now in grad school, I've worked with C. elegans, which are these little worms, and I've studied fertility in those guys. I've looked at zebrafish and streptococcus infection. And then I got to Jackson Labs with neuromuscular disease and mice. And it's basically exactly what I've always wanted to do. I'm actually doing a gene therapy trial. And I know that like little me, like even little elementary school me would just be like, what? You did this?
You did it.
Yeah.
So awesome. Well, what's something maybe that you didn't know in elementary school or high school, that you think is really cool now that maybe you would want to tell yourself, then, or, another student that maybe is interested in science and has had that cool experience with their, great science teacher, but they're not exactly sure, is it appropriate for me to make the next step? Can I do it? Is it something I really want to do? What's something that, you could say that's like, yeah, definitely want to do it because it's so cool? Maybe that's a hard question.
I think like the number one thing I would want to tell younger me is to never stop questioning things and always try to add onto your story. Back in high school, I was a little arrogant. I thought, oh boy, I'm going to become a neuroscientist and know everything about the brain. But as soon as I hit undergrad and actually started really going into depth about the research and what it took, I realized there's so much we don't know. And even though that can be a bit intimidating, that's also awesome. Like you can conduct those experiments. You can try to make that technology. You can try to ask others and make those connections. It's like a wild frontier.
That's
so
great.
I would just keep encouraging that, always asking questions.
So
And
be
even,
empowered by the unknown. Don't be afraid or sort of stifled by it.
yeah, definitely. Like, even like, so the reason why I have Crowley, the gecko, is because my landlord wouldn't let me have a cat. So I tried to find something that could fit in a terrarium that wasn't betta fish. I used to have betta fish in undergrad that I trained to jump through hoops and stuff like that. But I was kind of daunted. I'm like, I've never had a reptile before. How do I take care of this? And I had to like look it up, ask people questions. And now he's my little buddy, and I make him wear tiny outfits and do little photo shoots of tiny kitchens. And um, and that kind of goes on to like my philosophy too, of always try to like find something that adds onto your story, whether it's your science or even just your life. So for science, it's like, okay, what experiment will really fill out this story? What will answer this question? And in my life, I always think, what sort of opportunity can I take to meet new friends or have new opportunities? Like I went to a parking lot in the middle of the night one day to buy an emu egg off of a person on Facebook. Maybe not the wisest decision to just meet up with strangers in the middle of the night in parking lots, but I'm here.
Cool egg.
So
I like that.
It worked out.
Sounds like that might be one of your favorite parts of being an experimental biologist. Well, we have a question that came in from one of our viewers, Alana. She says, good morning to you. And she says, you mentioned gene therapy. Could you please talk a little bit more about that? Specifically, how do you study it in mice? Maybe we could just touch on that for a couple minutes. That's a great question.
Yeah, so the gene therapy I'm working on, so the disease that I'm using it for is spinal muscular atrophy or respiratory distress. It's caused by homozygous deleterious mutations in IGH-MBB2, which basically means if you have two bad copies of that gene, you're going to have SMARD or some spectrum of that disease, like more severe or less severe. And it's a good candidate for gene therapy because if it was like a dominant mutation, so it gained some sort of toxic function, you'd have to do some sort of therapy that knocks down that bad copy of the gene and introduce the good copy of the gene. So if this gene therapy, I just have to introduce at least one good copy for the disease to be mitigated. So I use an adeno-associated virus and I inject it into the cerebral spinal fluid, like through the brain, to these mice at day one, because it's the nervous tissue that's mostly impacted in these mice. So if we can get that directly, that gives for a better prognosis. And it's been going super well, and that's from a collaboration with Nationwide Children's Hospital in Ohio. So just because even if you two choose like a certain lab, it doesn't mean you can't ask questions from other fields or collaborate with other people.
I think that's a really important point that I learned is all of these extensive collaborations that come about, and you could never even imagine all of them.
Like we're even working with, so like at a poster session, I was talking about how with our mice, it's really hard trying to analyze respiratory distress. It's something you can see in the human patients, but it's kind of hard to tell a mouse to breathe as or exhale as hard as they can into like a little bag to see tidal volume and breath rate and stuff like that. So someone who was working with another company that looks at like sleep and respiration and breath rate and stuff like that came to me, and now we're collaborating, and they're from Kentucky, and we're trying to use their machines. Like they're originally used for just studying sleep patterns, but they do that through breathing. So we're thinking we might be able to use that aspect for our mice, including our gene therapy mice, so we can see that respiratory distress that we had a hard time like seeing before.
Wow, that's really cool. Well, I think that we're about ready to wrap up here, unfortunately. I could talk all day with you, Sarah. It's fun hearing about your path. maybe we could just touch about graduate school just for a moment. It's kind of an interesting and unique kind of school where you get a graduate school stipend. So you're actually getting paid to go to school a little bit.
Yeah. So I'm a first generation college student, first of all. Like, my mom was wicked proud that I just graduated high school. So completing undergrad was icing on top of the cake, and grad school, much like most of science, was just unknown territory. And the fact that I could be paid to go to school was fantastic. That's not always the case, like when it comes to some grad schools, like if you're going for an art major or trying to get a master's degree for teaching. But in a lot of STEM fields, you can get a stipend. And with the program I'm in, GSBSE, or the Graduate School of Biomedical Sciences and Engineering, my first year was just rotating with a bunch of different labs. That's how I got to work with nematodes and zebrafish and mice. And I got to get a little taste of everything. So
cool. Well, thank you so much for sharing a little bit about your journey here and your experience in graduate school. And thank you so much for bringing Crowley with so that we could hang out with him today too.
Yeah, he needed to go out for his morning stroll. So
maybe I'll just say thank you to you so much, Sarah, and thank you to our viewers, and ask you if you maybe have a final send-off message that you'd like to share with them.
Yeah, just never stop making stories. And if you have any questions, you can follow me on Twitter, which is SEHROTONIN, or serotonin. You can also follow the Cox Lab Twitter. Or
Sarah, we have one more question that came in from one of our viewers. Roberto wants to know, what are your plans after getting your PhD?
I still have a hard time thinking about that.
I know that's a hard question.
I first joined grad school partially because I'm like, I've known nothing but school for like the past 2 decades. I might as well continue. But I think the job I want to get into is probably like lab manager. I'm not sure if I want to go full PI because I still want to be able to really work in the wet lab, work with the tissue, see it directly, and coordinate projects, which you can do as a PI. But as a PI, I feel like your duties are more like writing grants and getting funding. But we'll see as the years go by. We'll see what opportunities come. Like I said, the way I've kind of lived my life is go with the flow and see what comes. So.
Well, I think that sounds like a good plan, and I'm confident that the flow will take you in an excellent direction. Really nice chatting with you this morning, Sarah. Thank you so much for your time, and thank you to our viewers. And don't forget to join us next time for another JAX Career Chats. Goodbye for now.
Thank you.